Hello, my name's Andrea, and in this presentation I will be talking about next-generation sequencing in acute myeloid leukemia. We will go over some of the background of next-generation sequencing and how this molecular technique works and how it can be applied to this cancer. In this slide I will be talking about the history and background of next-generation sequencing, which you will also hear me refer to as NGS. DNA sequencing is the process of determining the exact order of DNA bases. This was first done in 1977 by Frederick Sanger, who developed the Sanger sequencing method and also received the Nobel Prize for it. We now refer to this as traditional sequencing method, which entailed the use of gel electrophoresis to read a DNA-based sequence, the use of deoxyribonucleotide triphosphates, and enzymes such as DNA polymerase. Some of the major differences between the sequencing back then, traditional sequencing, and the newest technology is that prior knowledge of the genome was required to design PCR and sequencing primers, whereas now no prior knowledge is needed because the PCR and sequencing primers are universal. Moving on, in 1990, the Human Genome Project was started. Their main goal was to have the entire human genome sequenced, and in 2000 they finished their rough draft. Another major thing happened in 2000, which was that Lynx Therapeutics company launched the first of NGS technologies, which was massive parallel signature sequencing. Another company called Illumina eventually bought this new technology. In 2014, Illumina launched a high-seq extend sequencer and essentially monopolized the industry by holding 70% of the market for DNA sequencers and accounting for over 90% of all the DNA data produced globally. Back then, sequencing was extremely expensive and time-consuming, and now you have Ultima Genomics announcing that the genome can be sequenced for up to $100, and Illumina is also promising to generate more than 20,000 whole genomes per year, which is way more than we have ever before. In this slide, I want to talk about the Illumina sequencing technology that is used currently. There are 4 major steps, which are library preparation, cluster generation, sequencing, and data analysis. Library preparation requires for your starting material to be extracted and fragmented. Adapters are added, which are small synthesized pieces of DNA or oligonucleotides. The adapters are then added to each DNA fragment to make libraries. Each complete library will have the adapter and unique DNA insert. The adapters contain the binding regions for clustering that are complementary to the oligonucleotides in the flow cells. Flow cells are glass slides with fluidic channels where the sequencing chemistry occurs. For cluster generation, the libraries are hybridized onto the flow cell and then are amplified through a process called bridge amplification. The result is a distinct clonal cluster for each fragment on the flow cell. Then we have the actual sequencing part, which requires fluorescently labeled nucleotides added. They bind to the complementary DNA bases in the clusters, and the instrument images the flow cell. The image information identifies the first base in the cluster. The process repeats to determine the sequence of the bases for the clusters in the flow cell, one base at a time. When the flow cell is imaged, each of the bases emits an intensity of a unique wavelength that makes it identifiable. Finally, data analysis occurs after the base calling is complete. This includes alignment to a reference genome or variant detection. Data visualization tools transfer sequencing data into various formats to allow you to explore and analyze data from one or many sequencing runs. For the equipment needed, we will need standard laboratory equipment like gloves, coat, goggles. Other equipment can be heat blocks, centrifuges, vortex, pipettes, pipette tips. We would need extraction equipment, which could be either done with manual extraction methods or automated platforms. A thermal cycler for the bridge amplification step, quantification equipment to quantify how much DNA or RNA is in our sample, and of course the sequencers. There are various sequencers out there. The ones shown in the pictures below are created by Illumina, and the ones on the right are done by Oxford Nanopore Technologies. The things that you would need for the sequencer for the most part are reagent kits, which include the flow cell, and the cartridge, which also allows for the chemistry where the sequencing chemistry happens as well. And library prep kits that contain all your reagents that you would need to prepare your sample, like DNA and RNA indexes, which are your adapters, fragmentation beads to fragment your sample, beads, and buffer. Acute myeloid leukemia can be abbreviated as AML and is a type of cancer of the blood and bone marrow. It affects a group of white blood cells that are called the myeloid cells. These cells normally develop into various types of mature blood cells such as red blood cells, white blood cells, and platelets. You can see symptoms that mimic those of the flu or other common diseases. They can be fever, bone pain, fatigue, shortness of breath, pale skin, frequent infections, unusual bleeding, and a few others. Some of the major diagnosis tests that can be done are blood tests, bone marrow tests, and testing for cancer cells with lumbar punctures. Typically, we see too many white blood cells, not enough red blood cells, and a presence of blast cells in the marrow but not in the blood, and a lot of gene mutations. A complex multi-gene sequence of acquired mutation drives most of AML cases. This can also be treated with non-specific and highly toxic chemotherapeutic drugs like the ones shown here, cytarabine and anthracycline. The diagnosis of acute myeloid leukemia is based on the morphologic, immunohistochemical, and flow cytometric identification of the myeloid blast cell. AML also has subclassifications that are defined by a specific gene mutation. Conventional subclassifications have limitations, though, and are now supplemented by more clinically relevant classification schemes that are based on previously mentioned lab techniques and molecular genetic information. The majority of AML diagnoses are defined by a specific chromosomal translocation in the chromosome 8 and 21, and as well as the presence of pathogenic mutations in any of the three genes that you can see here, NPM1, CEBPA, RUNX1. These define a specific diagnostic entity in the current World Health Organization system. Diagnoses defining chromosomal alterations are usually detected by either cytogenetic techniques such as karyotyping or fluorescent in situ hybridization. However, newer technologies such as NGS-based fusion gene panels are being increasingly utilized in the routine clinical diagnostic laboratory to detect the same and many other chromosomal translocation events. The detection of NPM1 and CEBPA mutations has been integrated into the initial diagnostic workup of AML for many years now. Up to more recently, the RUNX1 mutation detection was also integrated into the initial diagnostic workup of AML. Because of the increasing availability of NGS, this new method is quickly becoming the gold standard for detecting AML mutations that are defined by the World Health Organization. NGS plays a major role in the prognosis of acute myeloid leukemia. Although the World Health Organization has already defined these three genes, NPM1, CEBPA, and RUNX1, as to have significant prognostic relevance, the NGS technology can be used to target those three mutations as well as many others. AML has been shown to contain multiple pathogenic driver mutations per tumor, and when doing whole genome sequencing, even a larger number of total somatic mutations are identified. These mutations have been shown to influence long-term AML prognosis and provides practical AML risk stratification information that include both large-scale chromosomal alterations and small-scale gene mutations that are currently not distinct diagnostic entities based on the current classification system. These prognosis-defining mutations have been shown to predict a specific AML disease outcome based on the presence and absence of certain co-mutations. Co-occurring mutations in AML is a significant determinant of a patient's response to induction chemotherapy. Some patients can be more resistant to cytotoxic chemotherapy than others, and it essentially influences the standard of treatment in those decisions. It also allows for family members to be evaluated for germline mutations depending on if the patient has the gene or not. And as you can see here, you have certain genes like NPM1 and CEBPA. They confer a good prognostic outlook, whereas having the RUNX1 gene confers a worse prognostic outlook. These are some of the other applications that you can see with NGS technology. Obviously, we have cancer research where you can have targeted sequencing for oncology. You can also have liquid biopsies in drug and companion diagnostic development. It can be used for complex disease research that targets gene expression, epigenetics, and methylation analysis, of course, whole genome sequencing, and targeted DNA and RNA sequencing. You can also have infectious disease that does bacterial, microbial, and viral typing and sequencing. For reproductive health, you can have expanded carrier screening and pre-implantation genetic testing. For human identification, you can have forensic DNA analysis and phenotyping with NGS-based technologies. Agrigenomics focuses on targeted genotyping by sequencing on plants and animals, and HLA typing, that is NGS-based genotyping class 1 and class 2 loci. For the potential future, we already know that NGS-based assays are targeting a panel of consensus gene mutations associated with MLA. They have become increasingly available in the clinical diagnostic laboratories over the last few years, and enhanced NGS-based assays and bioinformatics tools can capture not only the short DNA sequence mutations, but can also copy number variants, as well as AML-related chromosomal translocations via RNA sequencing. These are providing time-sensitive and cost-effective comprehensive mutation profiling for AML patients. However, some of the things that we can improve in the future are simplifying the technical complexity of this technology, because it is rather complex and a lot of training needs to be done before a technician can use this type of technology. Improving turnaround times would also be ideal, and reducing the need for specialized and high-level expertise required. It is an intricate job to be analyzing sequences and doing it in the right way. And of course, I think the ultimate goal is to reduce the cost even more for sequencing, so it can be done also in low-income countries. In summary, today we talked about the background of next-generation sequencing, some of the previous sequencing techniques, and how it has developed to NGS. We also talked about the molecular nature of AML and its clinical and phenotypical manifestations, as well as the diagnosis and prognosis of AML using next-generation sequencing technology. We talked about other current NGS applications and some expectations for the future. When it comes to other current NGS applications, I also want to mention that it can also be used for surveillance purposes and for things that you don't necessarily need to have prior knowledge of a genome, such as wastewater testing, where you could have a various diversity of pathogens, bacteria, and so on, and NGS really allows you to sequence wastewater in a way that it gives you a potential fit for what could be found in those water reservoirs that can be very helpful for public health. And that is the end of the presentation. Thank you for listening today.